What's up, Denmark? Hi. So yeah, I don't really need to introduce myself very much then. I've been working on varroa resistance for pretty much my entire career, which has been about 7 years, not 40. So I have a little less experience, but I've done I got a chance to research some really cool things. So I'm gonna share those with you. I'll outline the talk first. So yeah, all the slides are in Norwegian. I put them there. It took me like half a day. So you can read them, hopefully, but I can't. So hopefully I've memorized enough that I can give the talk without much of a problem. So I'll talk a little bit about who I am. I'll talk about the first population of resistant bees that I got to study, which is a buckfast population in kind of South Norway. I'll talk about the new population that I get to work on that is part of a breeding program, and that's really cool. So we'll get into that a little bit. And then a lot of people ask me, well, if I want to get into resistance breeding, what should I do? What should I think about? So I'm going to ask a question. How many people are actually interested in varroa resistance breeding? You can put your hand up. Oh, so many. So cool. All right, you're going to like this. Here. This is me, the person, not the cat. The cat is Celeste. She's very sweet. I'm originally from Canada. I did my PhD at the University of Bern, the Liebefeld Institute of Bee Health in Switzerland. And I started right on varroa resistance. I knew nothing about bees before I started my PhD. I had about 6 months over the winter to learn everything I could. So I took as many papers as I could find and read them all and built my knowledge. So yeah, I've been researching varroa resistance for about 7 years. And now they put me in charge of a varroa resistance breeding program in Hurdal. So that is that's me, what I'm doing. I get to skip this slide because Stephen's presentation on varroa was so good. You know everything there is to know. So I'm not going to talk about varroa here at all. We'll go straight into the buckfast population that I got to research first. So I think about 1995, varroa started coming into Norway, and it hit everybody, and it hit everybody bad. So in 1998, a guy called Tarje Reinertsen decided that he was going to stop treating his bees. You know, his logic is like the acaricide, the stuff that they were using to treat, they were using doses for different countries, and it was really potent in Norway, so it was damaging a lot of colonies. So he's like, well, either the mites kill my bees or I kill my bees. I don't want to kill my bees, so I'm just going to leave it. And he did lose a lot of colonies at the beginning, cutting it cold turkey, but not all of them. So some of them survived, and he bred from those and worked his stock back to a functioning level. And now these colonies are really big, they are very gentle, and they produce a lot of honey. And they have been completely untreated since 1998. Nothing, not even drone cutting. Absolutely nothing is done to these bees. And they seem to be just fine. The first data was taken a year before I started in 2014. And I have that data. It's a bit of virus data. I can talk about it later, but it's not in this presentation here. What we're going to continue on. Right, terminology. So I'm going to talk a little bit about the terminology because there are words that are used that have different meanings for people. So these are the meanings in these talks. We have varroa resistance, and to me and what I'm working with, that means the bees are actively reducing the population of varroa. So they're taking them down, they're controlling them, so there's less varroa in a colony. Something like tolerance, the bees are less good at controlling the varroa, but they don't seem to be dying. And as in Stephen's talk, this could be from different reasons, like the fact that there's no viruses in the system, so the mites have less of an effect. But it could also mean that there's some mechanism that bees have that resist viruses we don't know yet. But the bees that I'm going to be talking about are resistant. They can reduce the number of varroa in their hives. Either tolerant or resistant, all bees are surviving. A lot of the graphs you're going to see, I have surviving bees and susceptible bees, and that's a scientific differentiation because we weren't sure which it was yet, tolerance or resistance, but we're pretty sure now that it's resistance. But they're surviving. They can live, with varroa infestation, and they're okay. So, excuse me. What I did was I took a lot of data over a lot of different years from the buckfast population. A lot of these were bottom board counts. So basically you shove a board under the colony, you leave it for a week, you count mites, you replace the board, you do it again, and you take 2 or 3 different samples, so you get you get a pretty sure number of how many mites are falling from the colonies. And right away, the surviving colonies are going to be in blue for the entire talk, and the susceptible colonies, the control colonies, the ones that need to be treated, they are in orange for the whole talk. So you can see right away, the blue bars are lower than the orange bars, and all of these pretty much are in August, so the critical period when the colonies are making winter bees, when you really don't want varroa in your hives, this is the levels. So they're always lower than the susceptible hives. And this is consistent year to year, 2015, 2017, 2020. If it was just 1 year, I'd be like, maybe it's a fluke, something environmental, location, I don't know. But they've done it again and again and again, and they're still doing it. So this is pretty good evidence that there's something going on. Right, let's take a closer look. I did an experiment where I basically just watched 25 hives in each of these groups, like a hawk, and I had bottom boards all the time. So every 7 days I went out to the hives, I counted these boards, all 25 of them in one and all 24 of them in the other, replaced the paper, put them back in, and I did this from the middle of May until the middle of August. So every week there was an average of mite drop in these colonies. And as you can see, the orange line pretty much did what we expected it to do, in that around week 6 or 7 in the study, it just started going up and it didn't stop. This is pretty classical for mite infestations for a colony that has no resistance. But if you take a look at the buckfast line, the blue line doesn't really seem to have a problem. It goes up a little bit, but it doesn't really move. So the loss rates are generally pretty low. So this is what's happening over an entire season, is the mites aren't increasing more than about 5 a day. And this is fairly consistent. So this is what a surviving population looks like for an entire season. The mite levels, the drops on the boards just don't go up. So what's happening here? In 2017, a paper was published, and I think the work was done in 2015. So in order to kind of tease apart the effects, and because the buckfast survivor bees didn't have any mites, what I did was I took frames of brood from colonies that did have mites, 2 from each colony, and one of those frames I put in a survivor colony, and one of those frames I put in a susceptible colony, just after capping. And for 9 days in the period of the capping, we left them in the hives. And then I took them out and I dissected them and counted how many mite offspring they each had. So Stephen said that the general offspring rate is 1.4, right? 1.34, something like that. The susceptible colonies, which are actually now in dark blue, that's wrong, I apologize for that, that should be orange, are around 1.24. But with the standard deviation, the error rates, you could probably, it's pushing 1.4. It's probably not very significantly different from 1.4. But in the buckfast survivors, 0.87, less than 1. And now the brood had come from the same place, and the mites had come from the same place. So they were all the same. The only difference was the 9 days in the survivor colony or the 9 days spent in the susceptible colony. So this told me that the worker bees in the surviving colonies were doing something to the brood frame to reduce the number of mites that were being produced. Right, what's happening? Breaking it down a little, this is the mite fall again in August, but by the proportion of the colonies that were there. So we have the number of mites falling on the boards daily, and then you have the proportion of the population, and this is in August. So 80% of the surviving bees have between 0 and 10 mite fall per day. And there's some colonies that are a little higher, but most of them are between 0 and 10. The controls, however, at the same time, a much wider spread, and you've got a lot of these colonies who are just really loaded with mites. Those colonies you do not want in your population. So, knowing all of that stuff, and I'm getting to recapping, don't worry. I was asked to design a breeding program in a little valley called Herdal. And the goal of this project was basically to recreate what Tarja had done, but capture it all in data and write it all down so that we could come up with a recipe that anyone could follow to start breeding for resistant bees. I'll introduce you to Blue 42. She was one of the first queens produced in the breeding program. She's my favorite queen, not just because I have a picture of her, but because she was scored one of the top scorers on the algorithm that I used to basically score the queens. 2 of her daughters also made it on to the top scoring the year after, and they've produced now the granddaughter variation, the yellow generation that is in the colonies now, and we get to test them next year. So she's really cool. Her recapping rate was something like 40%. With a 70% accuracy rate, and her mite levels were like 0.7. She's a really cool queen. I like her. And she's still going. Last year she was still there. So, what did we have? We had basically the perfect storm to start this experiment. We had an entire community of beekeepers that lived in the same area, worked bees in the same area, willing to stop treating and basically join this program. We had a queen breeder that lived in the valley as well. And the really cool thing about him and his bees is he had already not been treating for about 10 years. So we had a head start. We had a population of his bees that we could now see the entire valley with that had not been treated for 10 years. We also had this valley, which was, apart from the beekeepers in the valley, there is not a lot of colony activity going on around it. It's surrounded by mountains. It's backed up against a lake. So it is fairly, in theory, isolated. I'm sure there's crossover, and not all of the beekeepers in the valley buy queens from our queen breeder. But we have an incentive program to make sure that the people at least involved in the project buy queens from him. And so we had all this, and now we had a profile of a really strong surviving population, the buckfast bees. Now the buckfast bees, they live close by, but this is in a conservation zone that keeps only Carnica stock bees. So there's a wall there. No genetic material gets in or out of this place, in theory, that isn't Carnica. So we had a kind of a barrier there. And we started in 2020, and we're continuing. So we have about 3 years of data. We have about 2 years of selection going on, and I'm going to show you what's what there. This is the type of data that we took. So we had a little over 100 colonies in this valley, and the beekeepers agreed to take data 3 times a year, in spring, in summer, and in fall, and then also the honey harvest data. So I asked them to count mites 3 times a year, and to try and count more than once, so you get, this kind of couple of weeks of data. I took colony strength on spring, summer, fall, the status, general health, colony ID, or queen ID, so that we knew which queen was in which hive where. We had a general health measurement, which was 1 to 4, and temperament, very important for beekeeping, also 1 to 4. And then, so the beekeepers were taking that, and I was taking about brood samples from about 10 different colonies. It's about 10% of the population, and then measuring not only mite metrics, but also hygienic behavior metrics as well. Right, so this is the first stuff. This is the average daily drop from 2020 in light green, 2021 in medium green, and 2022 in dark green. We have the number of mites falling per day in spring and summer and autumn. Right away you can see there's no great drop between 2020 and 2022. They seem to be fairly stable. But the interesting thing is that they are stable. This is a population that has a lot of colonies that have not been treated for a long time, and that you really see the effect of that here. There's some colonies that got completely outrageous, but the general trend is not exponential like we saw with the orange line on the graph before, but it's kind of like a steady increase, and it seems to reset back to 5 every year, which is very interesting. And then the general colony size, nothing really that special to report. It looked like in 2021 the colonies were a little smaller in autumn, and that affected the spring colony size, but it didn't really carry over into summer. The bees kind of brought themselves back up. This is the same kind of bar graph that shows in August how many mites were falling per day over the years. And so between, I think 0 is like 0, 0, and 10 is like 0 to 10-ish. So it's 40–45% of the colonies are in that golden zone, which is really good. But you still have that tail of outrageous colonies that are probably seeding mites into other colonies and causing trouble. So if I put this line, the light green line, this is kind of a, I think this is 2021, and I put it against the survivors and the susceptibles. You can see it's kind of smack in the middle of both of them. So we're doing pretty good, but we're not quite there. The goal of this was to try and manipulate the bees so that we could get them into the blue lines and take away all of the ones that were mimicking the orange lines here. So we're kind of, like we started in a really good place, and we're going. So now let's talk about some interesting recapping stuff. So this is what basically ultimate what I say is that the gold standard for beekeepers who want to breed for resistance, if you're counting on bottom boards, you want to try and get the colonies that can keep it under 0 to 10 in the critical period in August, and you want to get rid of the colonies that can't keep it together. So either by drone cutting or treating and just removing the queen from your population, requeening, or putting it aside and requeening it next season. This is kind of what you want to focus on. So mite reproduction. There was a little bit of a difference in 2022. So 2020 and 2021, I can't say those are very different at all, but from 2021 to 2022, that might actually be a trend. It might also be luck. 1 year, I never say, is worth much of anything. It needs to happen more than once. I don't think it might be statistically different from 2020. But this is only 2 years of real selection. So I'm not expecting the entire world to change in 2 years. I predicted about 5 of selection, because we also have a free mating system. We're not controlling the drones at all. The drones can come from wherever. Colony losses. Not much difference. It's not great, but for a population that has not been treated at all, it's not bad. But we can do better. I think we can probably get this down with the right selection. Let me figure out what's going on. Right. How much time do I have left? Oh, lots. Yeah, great. This will be fine. Cool. All right. This is probably what people came here to see, and this is cell recapping. So I'll go over this paper. I go over it almost every talk, but this is the thing that really struck me. So I was sent to Norway to look at varroa-sensitive hygiene, so brood removal, and I was sent to look at grooming. So I set up the experiment to look at those things, and I found none of them. Absolutely none of them. They were not removing brood at any greater rate than the controls that I had set up. They were not grooming mites off at any greater rate than the controls that I had set up. So I was sitting there, I think at about 11 o'clock at night in the lab, questioning my entire life, being like, what am I doing with this PhD? I've wasted an entire year and I have nothing. I had read a paper previously on a behavior called cell recapping that a lot of researchers had kind of dismissed as a byproduct to brood removal. And so I didn't think much of it because I was, well, there's no brood removal. I'm not going to see. But then I actually look at Tarius frames. And I noticed something, and one of the caps looks a little weird. I'm like, nah. So I very carefully open it, turn it over, and there's a hole in the cell cap. And how you can tell, it's sometimes very difficult to see from the top, but picture A there is the top of a recapped cell. It looks a little different. Now some cells are just depressed, and then if you open them, there's still the silk cocoon there from the pupa, and they're not recapped at all. So the underside would look like picture B. So the pupa, when it spins its silk cocoon, it's very glossy, so you turn the thing over and you can see it. Also, the wax is the same color throughout. If the cell has been recapped, the bee has chewed a hole in it, you know, looked in, thought everything was fine, and put the wax back. So the silk cocoon is gone, the shiny thing is gone, and you can see darker wax filling the hole. I don't know if you can see it on the slide. Can you see it on the slide? Good? Okay. So that's what I found that at 11 o'clock at night. And I was like, oh, wow, that's interesting. And the next one was recapped, and the next one, and the next one. That one not, but the next one was. And sure enough, this recapping was everywhere, but only on Tarius frames. Again, the brood had come from the same place. The bees, the larvae, the mites, all from the same place. What was different was the bees in the buckfast hives. So taking a look at the recapping, I went back to my supervisor and I was like, we have 2 other populations of surviving bees in Sweden and France. They published on it. I know, I read it. I spent 6 months. But so can we ask them to test it, look for it in their colonies? And they did. They all did. And this is what they found. So you can't read this. It's ridiculously small, but I will read it to you. The dark black bars, these are the surviving colonies here. And each population is divided by these dotted lines. These 2 groups here are cells that were dissected that had mites in, that were infested, and cells that were dissected that had no mites in, that were uninfested. And then this is the proportion of recapping that was going on in the brood samples that we took. And right away you can see that all of the dark black bars much higher than the grays, which were control colonies, but in the same countries, in the same areas as these survivors. And the black bars were the highest in infested cells. So not only were the surviving colonies doing this behavior much more frequently than controls, but they were doing it on infested cells much more. They were targeting varroa. Huh. That's pretty cool. And since I've done this work, then Stephen and his team have done a lot more work, and they found recapping in pretty much every population that's been tested for it. So now we know that this behavior is very highly associated with colonies that can survive mites. And it's easy to see. Once you train your eye to it, you can look at a frame if it's a strong survivor, and you can see it just by looking at the frame. Cool. So there's a couple there's types of variation in this trait. And I've broken it down. This is my way of explaining it. There might be other ways, but there's two different components to this recapping trait. There's the frequency, so how often they recap cells, and there's the accuracy, how often they get it right. So you can have colonies that recap pretty much everything, but they're hitting mites randomly by accident. And you can also have colonies that don't do a lot of recapping, but every cell they recap has a mite in it. So they're really good at getting it right. And these two different types of variation, they can change in each colony. So they can be at different levels in each colony. I think they are independent, probably linked, but they can fluctuate on their own. Then there's another set of variation that makes my job very difficult, and that's environmental variation. So when there are a lot of mites in a colony that is a strong survivor, strong recapper, like say they go and rob something that's failing and they get a whole bunch of mites and the mites jump in the brood, they can turn on this behavior and then they go nuts and recap pretty much everything. And then you kind of see a lower reproductive rate in the mites in these cells. But the same colony that has no mites, the bees don't really think they need to do anything. So they're like, do, 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 do. And the mites that you do find, no recapping, and they can reproduce kind of as normal. So sometimes the bees need to be challenged before you see the trait. It's not going to be there all the time. So if you have a colony that isn't producing this trait, don't worry too much. If the mite levels are low, you're probably not going to see it. I mean, you don't clean your house when it's already clean. You clean your house when it's dirty, because that's extra work. Right, so taking a look at recapping. The top graph here is the proportion of recapped cells in the surviving buckfast bees and the control colonies that I used. That's the number of recapped cells in the brood. This down here is the accuracy, so the proportion of infested cells they managed to get with this behavior. So you can see both for the buckfast survivors are higher than the control type. It's not very high, but the accuracy is much higher than the frequency. So they're targeting, and this data is separate from the black and gray bars that I showed you. This is another year of data, so they're basically just doing the same thing this year as well. Very different from the controls. Now let's put Herdal on there, because this is the most interesting thing I have from the Herdal project so far. You can see on the frequency, not too interesting. It probably isn't very distinct from the control population. There's about 10% recapping, which means there's, something going on, but not a lot. But from 2020 to 2022, so when we started selecting actively, the accuracy shot up. And 2021 is kind of in the middle here. So it's just gone whoop. That could be something. That is very interesting. So the accuracy basically tripled in about 2 years. That is very fast. So that is pretty cool. And if it continues, we might just continue to see the drop in reproductive rate. And if that happens, that is really good evidence that recapping is very tightly associated with survivability and can be used as a selective trait to pick your survivors. But what I'm doing now is actually we're just selecting on bottom board counts, because what I want to do is create a method that's very easy that everyone can do to select for their bees. But I'm basically kind of tracking recapping too, just to see exactly how connected they are. So, okay, what did we learn? We learned that bees are not defenseless. None of them are, actually, because even in the control population, there were bees that were recapping at high rates. So there were colonies in there that had potential and could be bred from. I stand firm in the belief that pretty much any population of honeybees can be selected for resistance. All of them have a little bit of resistant in there somewhere. The colonies that did really well in surviving, they kept the mite loads stable throughout a year, almost almost completely stable. So there was no increase. So from spring to autumn, not a lot of increase at all. So you need to look at the trend, what they start with and what they end with, just as much as how many mites there are actually in the colony. The best survivors only had less than 10 mites a day in autumn, in the critical period. That's what we know. And we also find recapping is it's constant, it's trackable, it's observable, you can see it, and it exists in every surviving population we've tested for so far. So this is the summary slide. This is pretty much the information that I'm giving you here. So what can you do? The first thing I say, if you want to actually breed for resistance, you need to know how many mites are in your colonies. It's for the safety of your bees, the safety of other people's bees, and so you have a really good idea of which queens are going to be successful and which queens need help. So, you know, if you find queens that need help, that really aren't doing well, you can actually do something before you lose the hive, and then you just requeen her next year. It's very important you know how many mites are in your colonies. The next thing I would say is try breeding some of your own queens. Pick a queen that you really like. And if you don't have the means to breed yourself, you can make friends with a queen breeder or bring a community together and use the resources pooled together to start a smaller operation and try it and learn. Because these traits are quite apparent, and you will learn a lot just by trying to do it. Yeah, and my third point I already made is work together. The reason why the Herdal project worked so well is because the beekeepers in the same area came together and decided we are going to do something about this. And they're all working together. They're trained beautifully, so they collect such good data. And they're really great at working together. So I think that is pretty much the secret behind Herdal's success, is the beekeepers and how good they are. It's not me. I had some questions on, well, what's your selection protocol? What do you do? How do you pick your best queens? And I'm going to put a disclaimer right here now on the front, is I designed the algorithm that was the best for Herdal. It might not be the best for your area, for your bees, because everything is different. There are a lot of different bits of environmental variation, even over such a short distance, Norway to Denmark. You know, your landscapes are different, everything's different. So this is what I did. It's take it with a grain of salt. So for honey production, you're supposed to basically take the average for the apiary and then create a point system on how much higher or lower than the average each colony is. So I gave about 1 point per kilo. But the most honey that is ever produced in Herdal is about 50 kilos. If that's low for you, then you can change your point system to accommodate for that. The next trait was gentleness, how tame they are. And everything that did not get a perfect score got no points at all, because we did not want angry bees. It didn't matter if they were resistant. If they're not nice, nobody wants to work them. And this is the problem I have with a lot of stock that was bred by scientists, is they're like, oh, they're resistant. It's like, yeah, but nobody's going to touch them. They don't like them. And then the last one is the mites, and that was based on the under 10 number in August, basically. And the selection is they got a lot of points if they had under 10 in August. And if they had anything over 20, they got a really low score. So we're really trying to select for queens that can keep it in this optimal range. And we're trying to get rid of the queens that can't. And that is pretty much the end of my talk. So I want to thank the beekeepers, without whom none of this would actually be happening ever. I mean, the Research Council of Norway is the one funding the project, and I work for Norges Bjørkelag. So thank you very much.